Hi. During this laboratory practical, we will extract DNA from both strawberry and bananas.
For this laboratory practical, we will use a scale, 2 100 ml graduated cylinders, some ethanol, dishwashing liquid, pineapple juice, salt, distilled water, bananas, strawberries, clock glasses, and 2 mortars. DNA is contained in the nucleus of the fruit cells. In order to set it free, it is necessary to break open the cellular membranes. Since these membranes are composed of phospholipids, fatty molecules, we will dissolve them using liquid soap. We will also use some table salt, NaCl, which will neutralize the negative charges, facilitating the removal of the proteins around which the DNA is coiled up. We pour 10 ml of dishwashing liquid in each graduated cylinder.
Now we weigh 3 grams of salt on the scaler.
Soap acts on the membranes of the nucleus and of the cell itself by destroying them. Subsequently, we pour the 3 grams of salt in the cylinders. In the first one, and then in the second one. And then we add 100 milliliters of distilled water in each cylinder. In the first one, and then also in the second one. Sodium chloride, dissociated into its ions Na+ and Cl- acts on the histones by breaking the electrostatic bonds that divide the proteins from DNA. We mix until we dissolve the salt. The mixing has to be done very gently in order not to create bubbles. We weigh 100 grams of strawberries and 100 grams of banana. We mash the banana and the strawberries. We mash it until it's a liquid pulp. We let them rest for approximately 5 minutes. We weigh 100 grams of strawberries and 100 grams of banana. Then we pour each pulp into the beaker. And then we add the solutions. Now we mix. Now we let the solutions rest for 5 minutes. We are now filtering the banana pulp. We now filter the strawberry pulp. Depending on the type of fruit and its degree of ripeness, for equal mass of fruit pulp, different DNA quantities will be extracted.
We pour 25 milliliters of banana and of strawberry filtered solutions into 50 milliliters graduated cylinders. We add 5 milliliters of pineapple juice in each cylinder.
Pineapple juice starts protein digestion. That means their destruction down to amino acids. Pineapple juice contains bromelain, a substance that destroys proteins, particularly the histones which are bound to DNA. Using a pipette, we extract 10 milliliters of solution and we transfer it to a 20 milliliters vial. Now we pour 5 milliliters of cold ethanol in each vial, making sure that the pipette does not touch the solution. The DNA that was in a water solution is now in contact with ethanol. In this environment, DNA is non-soluble and becomes visible. DNA looks like a transparent jellyfish. A qualitative and a quantitative comparison of the DNA contained into two vials makes it possible to state that from an equal starting mass of the used pulp, there are clear differences. Therefore, from the same starting mass, strawberries have a larger quantity of DNA than bananas. Bananas have a triplet 3N set of chromosomes for many of its strains. From strawberries there exist many strains, crosses and hybrids. In order to obtain larger fruits, the chromosomal set has increased to 4M, 6N, 7N and 8N. Therefore, for the same starting mass, strawberries have a larger quantity of DNA than bananas.